Hey, this week we're going to dive into nerves and in particular action potentials. Sorry for the bootleg setup still. I'm not quite figured out my camera angles and exactly where I'm going to record these. Had a few comments from my students taking the mic a little bit. Absolutely roasting hot here today, 35 degrees. So I'm hiding inside in the shade. And let's just dive straight in. So this is going to start off with the standard level content, and then we'll build up and finish with like saltatory conduction and how an action potential propagates for the high level stuff. Here's a basic neuron. So I've got a few slides that we'll talk through, and then I've got one of my web sheets that you can also go and download from Biology by Bradford. And it's a little bit interactive with some practice questions at the end that you can have a go at that gives you some feedback. So here is a simple neuron. We've got our cell body, we've got a myelinated sheath, which speeds up the action potential, the nodes around VA. So these are the areas that will what we call depolarize. And then we've got the axon endings. Here's the three different types that we have. I don't know where I've got all these pictures from. So if anyone wants me to quote them, then feel free to reach out. I will get better at sketching out my own pictures in the future, but still a little bit bootleg. Here we've got sensory neuron. So you'll notice that the cell body is actually found in the middle. And here are some little sensors that are going to cause depolarization from contact. And here's our interneuron. And you can see that there is no myelination of this. And then here we can find our motor neuron where there is myelinated. And then that's connecting to a muscle. So here we've got our sarcomeres. What's nice is to think about when we look at a cell to cut it into cross section. It's got all the parts that you would familiarize yourself with normally. Like the nucleus, the mitochondria, like proteins are being synthesized. So it has all the components for this to happen here. And at the end, we've got our synapse. And this is where a neurotransmitter is going to be released and the action potential will carry on to the next neuron if a threshold is reached. We'll look at synaptic transmission in a different video. Now, sorry, super shiny today. It's just sweating like crazy. Now here is a reflex. So before we talk about an action potential itself, like here is a simple neural pathway that they've added into the IB curriculum now. And you might be given a diagram like this, and you want to be able to identify the interneuron. Here is our afferent neuron. So this is our sensory neuron, and the effector neuron here. So you can see there's two junctions where we have a chemical neurotransmitter passing across the synaptic cleft. So at that point, the mechanism there is like diffusion by which it's moving. It's the slowest point because this is electrochemical where it's going down the axon. And our simple pathway would be a stimulus. Then we have our receptor, which is happening here in the skin. And the sensory neuron is transmitting that signal to the relay neuron, which is found in the spinal cord. So this is a cross section of a spinal cord, and our motor neuron. So this is not anything that you have to think about. It's like if you touch a candle, you sit on a pin, you're going to respond without thinking. It's just coming from the central nervous system. And this is to minimize any damage. Because if you had to think, wait a minute, that's really hot, I need to put this down, you've burnt your hand. So this is the graph that confuses everyone. And what's quite nice to look at first is this PhET simulation. So this is from PhET, Colorado. Just loading up here. And you can find it yourself. And here is a cross section of an axon. If we zoom in a little bit, you can see we've got some leaking channels. We don't worry about those too much in IB. Like it's not included in any of the mark schemes for discussing them. But what is important is what is the difference between the circle and the diamond. So the circle are the sodium ions and the diamonds are potassium ions. You've got two different types of ions. Now, in order for an action potential to occur, we need to have a stimulus, and then we're going to have a change in the movement of these ions. Now, this graph's important because you can see we have a resting potential of around minus 70, sometimes minus 65. It depends on what you're looking at in terms of like your source. But you can see if we add on the graph here and I stimulate the neuron, you can see what we've got here is around minus 65. And then if I just let it finish, and then we can zoom back through. Now, when we go from minus 65, minus 70 up to like plus 30, you can see in this graph here, plus 40, once again, as long as it's in that ballpark, it's not too pernickety. It's just that different sources are referencing different averages. That's called depolarization. And then this section would be repolarization, where it's going back down. And you can see it actually overshoots. So we call this an overshoot or hyperpolarization. It's got a few different names. But the point is that it means that it's harder for an action potential to be generated again, and it stops the nerve impulse going in the wrong direction. Now, what I mean by that is at this point here, you can see I've got most of my sodium outside and most of my potassium inside. And that gives me this potential difference. So a bit of physics, it's like the difference in charge between two areas, and our potential difference is minus 65, minus 70 millivolts. Now, there's something called a threshold, which is really nicely demonstrated on my web sheet that you can go through. And here's our minus 55 threshold. So an action potential is all or nothing. It either happens or it doesn't happen. So if I have a stimulation that's below this threshold, nothing's going to happen. If I increase it to here, now we're going to have an action potential. You can see the next bit's very rapid. Like an anecdotal story is that actually my daughter has developed epilepsy in the last year. And one of the medications she takes actually increases this threshold, like it's going closer to zero. So her threshold potential maybe now is like minus 30, and it makes it harder for an action potential to occur because that overactive activity in the brain is what can cause a seizure, which would cause the convulsing and the rigidity that you can see in epileptic seizures. So she's got two types of medications. One of them increases the threshold to a higher state so that it stops the chance of a seizure. The other one actually increases the reuptake of neurotransmitters and stops too many of them accumulating. So it's really interesting. Of course, whenever I go to the doctor, I think I have an idea of what I'm talking about and I try and get involved in the conversation. But I always emphasize to them, of course, I'm not a doctor. I'm just able to understand, and I want to understand a bit better, especially when it's about my own kid. And then if we go back to this graph, so what's nice here is I can click through. So depolarization, that's this region here. That's where our sodium, which is outside, you can see, is moving in. So the sodium will move in. You can see it rushing through that, all the little orange dots going in. And they're voltage gated channels. So the reason they open, like we're maintaining a high concentration outside, they only open when that threshold's reached. And because there's a high concentration of sodium, they move down their concentration gradient. So this is an example of facilitated diffusion. It has to go through a channel, it's passive transport, energy is not involved, and it's moving down its concentration gradient. And then when we have repolarization on this side, here, the green ones are leaving, the potassium's leaving. So potassium is moving down its concentration gradient, moving from an area of high to low concentration. It's passive, it's facilitated, it moves through a channel, it's too big, it's a charged ion, so they can't move through the membrane. Pretty much like 99% of the time, they will not get through the membrane. Remember, only small non-polar molecules can pass through the membrane, like oxygen or carbon dioxide and nitrogen gas. And we overshoot. The reason why we're overshooting, sometimes called hyperpolarization, is to minimize the chance of another stimulation, to move it further away from that threshold. And then what we have happen is we go through the next stage. This is where it resets itself, our recovery phase, and that's where the sodium potassium pump comes in. So it moves the sodium out and the potassium in, and it's moving 3 sodium for 2 potassium. And then that's it. So we just want to memorize these different steps. It'd be one of the harder questions, like a long answer question in paper 2, 8 marks, 7 marks, explaining the steps of an action potential. And then for the higher level students, which we'll move on to now, it might ask you about the propagation, so the movement of an action potential along a axon. So here's our axons. Now there's two things that can affect the speed of conduction. That's the thickness, the diameter of the axon here. So if it's wider, ions pass easily to conduct the impulse. When it's larger, so if we have a larger diameter, they're able to move easier through that axon, through the lumen of it. And if it's thinner, there's more resistance, so it slows down. And if we have myelination, only very small regions have to become depolarized. So it's like it jumps from one to the next. So it's jumping from one node in RAM VA to the next. And the mechanism there is what we call saltatory conduction. Saltatory because sodium is part of salt, is a salt, and it's a local current. So once the sodium is moved in the area that's depolarized, it then diffuses down its concentration gradient and causes depolarization in the adjacent node of RAM VA, which are not present in this picture. So it's happening like this. This area here has now got a high concentration of sodium inside. They move internally down to the next region, causing depolarization there. So if you want to, here's a little fact, it moves 30 times faster via saltatory conduction. So when you're asked about propagation, there's two things, myelination, and then you can explain saltatory conduction. All right, so thanks for listening to this. And if you want to go and test yourself a little bit, at the end of my web sheet, which I'll link, I've got, I always have like a nice little mind map connecting it all together. So if you click on these elements, you can see how they are interacting with each other. And there's a little gap fill, and my favorite bit would be the multiple choice questions where you get feedback and you can reset it and have another go. So these are IB style questions that will help prepare you for any assessment. All right, thanks. Remember to share, like the video, and of course, subscribe, and I'll catch you soon.